Okay, this is where the logic comes in, and I expect you to be able to logically think about what would, if this will increase or decrease the ability for the histones to bind DNA, and also whether or not it's going to increase or decrease transcription itself. So this is acetylation. The R group is a methyl group, and then this would be the point of attachment. This right here would be the point of attachment. Okay, otherwise, if this is a longer carbon, then it's called acylation and not acetylation. And so if we have a lysine, so there's a lysine. Once again, I'm just going to draw the amine on it. So there's the lysine. Whenever the acetyl group gets added to it, it's going to look like this. We still have the nitrogen, but now it's gone from being an amine to an amide. Okay? So whenever the acetyl group is on there, is it going to be less likely or more likely to bind to DNA? Less likely, because it's gone from being positively charged to being neutral. Okay, it's still polar, but it's neutral now. So it's less likely to bind DNA. So will it increase transcription or decrease transcription? Transcription increases, because if the histone is not bound as tightly, other proteins can wiggle in. My term, that's not the scientific term, but other proteins can have access to the DNA, and so then transcription can't occur. Okay, so the enzymes that do that are called acetyltransferases, because they transfer acetyl groups. And so we have histone acetyltransferases. Sometimes they're abbreviated as HATs, hats. And then they are very specific for what lysine gets acetylated. Then there are another set of enzymes that remove the acetyl group whenever it's time to turn off transcription. And those are called histone deacetylases, or HDACs. I used to like to call them HDACs, but one group in my lab that I worked in, they worked on a specific, it was HDAC8, and they would get upset if we called it HDAC. I don't know why. But so the deacetylase removes it from the lysine, the acetylase, or I should say acetyltransferase, transfers it to the lysine.
So only positive amino acids will be acylated?
Well, I mean, for these transferases, they are specific for a lysine, or maybe two lysines. It's not even just every lysine, it's very, very specific.
Okay, so when you hear that a gene has been acylated, it's only talking about the lysine.
Well, if it's a gene, it's not a lysine because the gene is nucleotide, right? And so those could be acetylated, but they wouldn't be acetylated by a histone acetyltransferase. It would be by some nucleotide acetyltransferase. But yeah, it is an acetyl group that would be, and chances are it's going to be through. Well, it either has to be through a nitrogen or an oxygen. And so, okay. And this is where I'm going to lead. This is where I'm going to end up. This is where we're ending it for the day, if I can talk. So the next two modifications I actually just put on the same slide. One is methylation, and once again, we're talking here about lysines becoming methylated, and then phosphorylation, and in particular, especially serine. For histidines, it's serine that can be phosphorylated. You're going to find out later on, I don't know if it'll be this semester or the beginning of next semester, that phosphorylation can also happen quite frequently with tyrosine and even threonine, but especially tyrosine and serine are the two biggies that get phosphorylated quite frequently. Okay, so let's just scooch this on over. So now, if we think about it, what's really going to happen to this lysine if it gets methylated? And this could be with a methyltransferase. Some of the cofactors that are used by methyltransferases can include like those that are folic acid derived, or well, that's the big one, it's folic acid, and it usually comes from methionine. Okay, so if we have the lysine with its free amine, Okay, and it becomes methylated. What's that going to do? And by the way, not only can it be singly methylated, and for those who've had chemistry cancer, we've actually already talked about this. Well, actually, we did talk about it with respect to histones, but also with other proteins. But lysines can be not only singly methylated, but what else can happen? They can have 2 methyl groups added, 3 methyl groups added, and even in an extreme case, you could have 4. But usually, it's just 1, 2, or 3. And so that's why if it happens to be trimethylated, it's going to, let's say 4. I'm sorry, it could be 1, 2, or 3. It can't be 4 because otherwise the nitrogen is making 5 bonds. Because it's going to become either a primary, secondary, or tertiary amine. And so, for example, if it's dimethylated, it's going to look like this. Single methylation, it's going to have 1 methyl group and 1 hydrogen. So what's happening to this? Is it going to be more likely to make the histone bind to DNA or less likely to make the histone bind to DNA? All things otherwise considered the same. Less likely, because once again, you're no longer positively charged. Plus, this is bulky. Like, if you keep adding carbons with hydrogens on it, those are a lot bigger than just having a nitrogen with hydrogens on it. Now, if it's trimethylated, it is technically positively charged, but it's huge, because it's going to have, 9 hydrogens and 3 carbons, instead of just 3 hydrogens.
So if it had like 2 hydrogens and 1 methyl, could it still bind?
And maybe it just won't bind as tightly. Okay. Or it may even depend upon the site, the location. Okay, for example, the CpG islands and things like that. For those who already had chemistry cancer, kind of have an idea of what we're talking about. But just in broad generalities here for the histones, this usually means that it's going to bind less tightly. So will it typically increase transcription or decrease transcription?
If it's more tightly or less tightly?
If it's less tightly bound,
Increase.
it increases transcription. And what you're going to find out, actually, if you really research this, is it's very site-specific. Like, different regions get hypermethylated or hypomethylated, because DNA can also be methylated, then it can even help signify where transcription should occur. But right now, we're just talking about protein modification. And, so let me scooch that on up. If we look at serine, and I've got the picture there of phosphoserine. So this would be a phosphotransferase, which is also called a kinase if it's utilizing ATP or GTP, or a phosphorylase if it uses inorganic phosphate. Usually for these, we're talking about some type of kinase. But if it's serine, of course, I'm just going to draw the end to serine. CH2OH, that's what it's going to look like. And we have our kinase. And the opposite enzyme that usually takes off is called the phosphatase. This is what it's going to look like. See, of course the CH2 is the same. O P negative. Negative, that's a phosphate. So the phosphoserine, is it going to, once the serine's been phosphorylated, if it's a histone, will the histone bind more tightly or less tightly to DNA? Less tightly, and the reason being negatively charged. The DNA is already negatively charged, so then it's going to definitely be less tightly. So therefore, will transcription most likely increase or decrease? Most likely will increase because the histone's not bound as tightly, so it relaxes, and the polymerases can get on in there and get their job done. So that's the kind of logic that I would expect you to be able to walk through. And you should be able to draw a phosphoserine at this point because you're just adding a phosphate group onto where the alcohol had been. So instead of the hydrogen there, it's got the PO3. And this was just a summary, just to show once again how we've gone through all of these different modifications from acetylation to phosphorylation, methylation, ubiquitination. Now this one doesn't show any of the sumo, but you could also have sumoylation, things like that. and how these enzymes that do transfer these groups on there or remove them are usually very, very site-specific. Typically, they are not real promiscuous just because you wouldn't want to just go through and add acetyl groups to everything, because that would be bad, or phosphates to everything. Or to remove all the phosphates and things or all of the acetyl groups, because once again, you're going to need certain ones in different locations.
So up to this point, they have all increased.
Okay, and so there are different ways to monitor DNA structure. I mean, if you have all the time in the world, perhaps you could crystallize it, but that is very time consuming, or do NMR. But one way that's used, and it's rapid, is through, sometimes they call it thermal melting, or even, this one's UV, but you can also do circular dichroism, which is similar. But what happens is DNA, whenever it's helical, it absorbs at one certain level of, only so much of it can absorb the UV light, around 260 nanometers. However, if it's been denatured, just random coils, then it's going to be able to absorb even more because the nucleotides are more exposed. Because right when it's in the helical, it's kind of looks like stair steps, so they aren't as exposed to the UV light. And so this is a thermal melt. And so what they've done here is you just start, of course, UV, this is at 260 nanometers. If it's double helical, you keep adding temperature to it, and it starts to melt, or denature. And at some point in time, the absorption ceases because of it doesn't increase anymore, I should say, because the fact is it's completely denatured. And the halfway point, the inflection point for this is called the TM, the melting temperature. And based off of the structure of the DNA, and like, for example, how many G's and C's versus A's and T's, and you can also do TMs with RNA as well, it's just not as set necessarily. But it does affect the TM. If it's a really, really long piece of DNA, then it's going to have a much higher TM. If it has a lot of G's and C's in it versus A's and T's, is the TM going to be higher or lower? It's higher because remember, each G and C, if it's a canonical base pair, they make 3 hydrogen bonds. If it's A and T, they just make 2 hydrogen bonds. And so this is actually really important for whenever you're doing PCR and some molecular biology or genetics experiments, because you have to tinker with the PCR machine, and it's based off of the TMs of your primers versus the, because you don't want it to be too high or too low, because otherwise your PCR wouldn't be successful.
But wouldn't there be like a certain point of the UV light that would cause dimers?
Right, but that's at a different wavelength.
Okay.
Mm-hmm. Yeah, so that's why you would just put your spectrophotometer at that specific wavelength for it to do the normal spectroscopic absorbance, since it's an aromatic compound.
So it'd be like a safe UV?
Yeah, so this is at 260 nanometers versus, I don't know what one's best for, or at what level is best for forming thymine dimers. But yeah, 260, it just, and this isn't usually at, well, it depends on the way the machine is set up. It's not for long periods of time. And so, because you just have to shoot it briefly with the thing. Whereas, if it was at those wavelengths for long periods of time, then you do have to worry about cross-linking to occur. But what we would do because there are times that you can use cross-linking to your advantage to look for structural things. And so when we would do that is you would actually leave the UV light on it for like 30 minutes versus 30 seconds. Okay, so now we're to RNA, and this is the part that's definitely not in the book. I'm pretty sure it's not even in the most recent edition. It is much more complex than DNA in structure, and so why is that once again?
Because it can.
It has the extra hydroxyl group, and so what does that extra hydroxyl group allow it to do? Not only can it be reactive, but what else can it do? Hydrogen bonding. And so there's just many more degrees of freedom with respect to it versus a hydrogen. And so I do want you to know the differences between DNA and RNA, and also to logically discuss like factors that affect RNA structure. It's very similar to factors that affect protein structure. So what were some of the factors that can affect the structure of proteins or RNA for this matter? What's one just general environmental factor? Temperature. You heat it up, RNA will fall apart. Okay, or if you heat it up enough, it could even start to degrade itself. So what else is there?
pH.
pH. So once again, because it can make it even more, nucleophilic. Because if you remove that hydrogen off, if you go if you go above the pKa for it, then you're gonna take it to make it an O-, and so it can go and attack. Or just the fact that the pH itself can, and actually there are times that this is done naturally to where it will change its shape based off of pH and other environmental factors. So it was inactive at one set of conditions and becomes activated under different sets of conditions in vivo. What's another thing that you may not realize, but it's especially true, even more true probably for RNA than proteins?
Hydrophilic reactions.
Close. Like what are some things that help? Like what is all RNA is, I can't say, it has this one characteristic that not all proteins do. What's one characteristic if you're drawing the backbone of RNA?
Oh, it has directionality?
It's got directionality, but none of that. Negatively charged. So if it's negatively charged, what else it's very negatively charged. What else must be there? Positive charges. And so it's actually dependent upon what metal ions, the ionic strength and what metal ions are present. So you can actually have very different shapes and functionalities if it's magnesium there versus calcium or manganese or cadmium or any of these others. And we don't have an inorganic chemistry class here. Otherwise, you'd learn about like inner sphere versus outer sphere contacts and hard metals versus soft metals, and because it can affect the reactivity of the alcohol. Okay. And so that's why it's really much more dependent. Proteins can be dependent upon that, especially if they're highly charged, but RNA is very dependent upon ionic strength. Okay, so I want you to learn how to read these. So, like I said, it's got its own lingo. This is the hepatitis delta virus, or HDV. It's a ribozyme, technically, which is an enzyme it's made up solely of RNA. It's not very big. But just like proteins have their own structural vocabulary, like alpha helices and beta sheets, these can form helical regions. They don't really do beta sheets, but they can also form loops and really tight turns and stuff that proteins can't do. And so it's actually quite complex the way that the structure can be. So it's got primary, secondary, tertiary, and possibly quaternary structure, just like a protein. So what would be the primary structure for an RNA? It's just its sequence of nucleotides. And it goes 5' to 3', just like proteins go N to C. This is 5' to 3'. The first one gets to number 1, and then this goes on from there. So that'd be its primary structure. What would be examples of secondary structure? Like if it's a protein, what's the example of a protein secondary structure? Okay, but just what are some examples of it? Like beta sheets and alpha helices. These don't form sheets, but they do form helices and loops and things like that. And so we're talking about localized structure. Whereas a tertiary interaction in proteins, what was the tertiary structure for proteins? It's similar to secondary, but there is a difference. Pardon? Yeah, it's like the global ones. The entire how it comes in, because you can have different regions that are not close together in the primary sequence that are interacting, physically they are close together. So it's a global structure. And then quaternary is when you have multiple polypeptides or something coming together. Well, here for RNA, the tertiary structure is very important because since it's so flexible, it's much more flexible than a protein. Because remember, proteins have that peptide bond, which is almost double bond-like. Remember, there's not free rotation around it. We have free rotation around RNA. RNA is very flexible. As you can see in that little picture. It's making some sharp curves. And so what's I think is really cool is an RNA can take and form a helix from two parts that are far apart in the primary sequence. And I'll show you examples of what I mean by that. And so you can have something like some of the nucleotides at the beginning of the 5' end and nucleotides at the 3' end, and they can come together to form a helix. Okay, and so that's why it's a little bit, like its secondary to tertiary structure is a little bit more complicated. And you can have a quaternary structure, like it can bind to, proteins. It can bind multiple RNA sequences can come together to bind things, like in the ribosome. Okay, but there are there's lingo, because they don't. If you see in this little example here, see there are P's and J's. This one doesn't have an L, but sometimes you'll see an L on there too, and those all mean something, and they actually tell you a lot of information. So, if we just start, this is, let's find position 1. Position 1 here is right there. There's the 5' end, and if you notice, it goes up, and then it's got this. This is like a secondary structure map. This is the tertiary structure. So it goes up, and then it goes over here. There's 10, 15, 20. Goes over here, up there, up like this, down, back, around, and on out. So that's why its primary structure looks it's like a maze. So they give it lingos, and one is P. And a P means a paired region or a helix. They just call it paired because it's not always the same degree as like the alpha helix or something like that. And as we've already talked about, how the fact that you can have like these tetraplexes and weird things like that. So P means a helix. It's a paired region. And they number them. Like P1 is the very first paired region you come to from when you start the 5' end. So it starts here. We have this helix. And then you notice there's this J12. J means joining. So it tells you the order from 5' to 3', what helices it is joining. Because a lot of times the J's are catalytically active. That's where some of the chemistry will occur. And so, for example, this one's joining helix 1 to helix 2. And then see, here's helix 2. But they don't have to join contiguously like 1 and 2. So if we look down here, this little loop, joining region, it joins helix 4 to helix 2. Because it actually goes this way. So there's only 4 nucleotides there. But those 4 nucleotides are what joins helices 4 to helix 2. And then sometimes you'll see L, and L means a loop. And in particular, like for example, let me find. Yep, okay. Oh, let me move that. This right here would be a loop. These are loops that are at the end of a helix. Okay, not the joining part. It doesn't join two different things. And that loop number is given based off of what helix is on. So technically this one would be L4 because it's the loop that's on the end of helix 4. Okay. So let's look at another example. Oh, whoops, this one, I thought I had tRNA here. I want to go across to tRNA first. What are they even having class? Okay, so we're back to this because this is just one example. RNA is really, really flexible and it can do various chemical things, but it also has some really interesting properties. And this is one property that it has that they have discovered. And this is called a kissing structure. And it's 2 different RNAs. And then we've got blow it up. And this is a helix, like the P region, and this is a loop. And but the loop is crooked, and these 2 loops, it's like they call it kissing, but to me it almost looks like they're holding hands. Okay, and they're making bonds in between the 2, what is that, red and blue? And what's really interesting about this kissing complex and the tertiary structure between 2 different loops is that physicists really get a kick out of this too, but if you slowly pull on it, it's like silly putty. You know, it's got some flexibility and it stretches, whereas if you pull on it really fast, it breaks, it's brittle. And so it actually has like differences in tensile strength than what you do. So, I mean, it just goes to show like how its chemistry and even its physical properties are much more flexible than what you would expect. You know, you can't do this with DNA because of its lack of flexibility, and even with proteins, it's a little bit more complex because of the fact that they have that planar bond in the peptide. But yeah, that one's just really cool. That one's called the kissing complex. And I had already mentioned this once before, but remember the loops, many times those loops are just 4 nucleotides long because that's called a tetraloop. And that's one that you'll see over and over and over again in the sense that they're highly stable. And so usually they're not really, really big loops. They're just these little tiny ones that are at the end of the helix. Okay, now I'll show an example. I forgot to point that out on the RNAsP. Go back. All right, so this is the secondary structure of the RNA that I worked on. And I'm going to blow it up to where we can see it. It can have domains and motifs just like a protein. This one's actually got 2 different domains that can fold independently of each other. In fact, where that dark colored line is, you can cut it in 2 and they'll still fold and be calically active on its own. And just ignore this little bit right here, because what happened was we cloned this and we closed it up to where that way we can make point mutations inside the actual middle of the RNA. And so, but yeah, naturally those that are, green or whatever, those 6 nucleotides are naturally not there. But if you notice here, this one, if we start at the 5' end, there's P1 and P2, P3. But P4, this is what I was talking about before. This is P4. There's one side of P4. This is the other side of P4. And in 3 dimensions, they actually fold up to form a helix. So it does not have to be contiguous to each other in the primary sequence, because they can make long-range tertiary interactions since it's so flexible. And if you notice, sometimes the numbering gets a little odd, like P5.1, but there's no P6, and there's a P5. And the reason why is a lot of times they find one, I don't remember which pRNA was found first, but they find one, usually in bacteria or something, and then they do homology. They look for sequence alignments to base off the other RNAs from other species, and not every species has all the same helices. Some of them have an extra one. So if you looked at E. coli versus Bacillus subtilis, E. coli doesn't have, I'm pretty sure it's a 5.1, but Bacillus has that extra helix. Okay, and so, and it's just in between where E. coli's 5 and 6 would be, for example. And so that's why you get some of those that are weird numbered. And like, for example, this would be L15 right there at the end of helix P15, that big loop region. Okay, but I just wanted to point out the fact that, yeah, this is very, very complicated. And in 3 dimensions, whenever I was working on this, it's hard to crystallize RNA many times because of the fact that it's so flexible. And this one's really big. In fact, the person who discovered RNAsP won the Nobel Award in chemistry. So, because it was the first true ribozyme that was discovered. Of course, the ribosome was around before that, but they used to think it was protein that did the work. And the ribosome couldn't find out it's RNA. But this is the first one, and it's a true ribozyme in the sense that you can take all the proteins away from it. In bacteria and under non-physiological conditions, the RNA alone does all the chemistry. It doesn't even need its protein. Now, in vivo, it needs its protein to become specific for tRNA. Okay, but I wanted to show this because this is what it looks like. Since this point in time, they found a way to crystallize it. Some of the people that we had worked with in the past, they found a way to crystallize the entire thing now. And actually, the models and stuff that we worked on was very, very similar. And so it was really nice to be corroborated on what we hypothesized. Okay, so yeah, so for example, this here at the end, this L12, that is called a GNRA tetraloop. So these tetraloops are very stable. And if you notice, the first one many times is a G. The second letter, the second nucleotide is any nucleotide. The third is an R, so purine, so like an A or another, either an A or a G many times. And then the fourth one is an A. And it's because the kind of bonding that it can form is highly stable. And so that's how you can get some of these kissing complexes and things like that. So like, let's see, where's another tetraloop? Like here's another tetraloop, there's another tetraloop. So you see that kind of thing over and over and over again. And you can also get what's called a bulge, which is really literally just something that sticks out. Like this little bulge here is called a U-bulge. And it's very important. That's actually where the catalytic active site is. And so it's right there. No. This was our model, and like I haven't updated with the actual crystal structure. And so this dark, and this is the 3D, well technically the quaternary structure. So this dark color, that's the pRNA that we saw before. I can't remember which helix is which on here, but this would be the specificity domain. This is the catalytic domain. This little tiny thing right here is the protein, the P protein that's in vivo is required just to help hold the tRNA still, so to speak. And then this light colored is tRNA. You know, normally you guys are used to seeing tRNA look like what? What does tRNA kind of look like? Like the way that they like to draw it a lot of times, it kind of looks like a little, they call it the clover leaf form, because it looks like this. But that's the secondary structure. That's not the tertiary structure. Tertiary structure looks like the sub-N or the L or whatever that's drawn like there. Okay, so that's why I just wanted to show you how complex this can be, and that this is tRNA, and this is the RNA that I worked on. Okay, let's go back. We got. So we've already gone over how it's more complex. I do want you to know the differences between the DNA and the RNA, be able to talk about its nomenclature, like what the P means, L, J. We've already discussed factors, especially ionic strength, which is it's much more reliant on ionic strength than many of the other structures that we've talked about in the past. Okay, so we're going to go over many different types of RNA, specific types of RNA that you probably in genetics or biology, you probably just learned the big three. I don't even know if they cover any of the others, but tRNA, mRNA, rRNA. Do they cover anything else? Maybe in genetics now they may cover like microRNAs or something. And so, very briefly. And so, yep. And then we're gonna get all the way down here to the PIs and talk about Pee-wee eventually, but not today. We'll have to leave you on a little bit of a cliffhanger to wait for Pee-wee. Okay. And so tRNA stands for transfer RNA. And I do expect you probably know what it is. I also want you guys to know about the relative sizes, like anything special that these may or may not do. mRNA was, or is, it still is, it hasn't stopped. What is mRNA? Messenger RNA. rRNA is ribosomal. Now the MI in siRNA, what is the MI?
Micro?
That's micro. Does anyone know why sometimes some of them, and I put them together, sometimes some people argue that those are two separate classes, but the SI, silencing or small interfering RNA or silencing RNA. SN, well, SNRP is whenever an snRNA hooks up with a protein, because then it's called a ribonucleoprotein complex. We're actually going to get to that. But the S stands for small, and the N is nuclear. SNO RNA is, well, the S, small. NO is nucleolar. And then, of course, we're going to talk about Pee-wee, last but not least. And there are other various types, I'm sure, that are out there, but we're just going to be talking about some of these in greater detail. So this is the generic tRNA cloverleaf structure that most people see. I want you to know all the parts to this. This is a secondary structure. This is not what it looks like in 3 dimensions. We've already seen where it looks kind of like the number 7 or the letter L, or for those of you that know your Greek alphabet, what is that, the lambda? No, gamma, isn't it gamma, like the capital gamma?
So whoever's in physics.
So, things like that. I always just thought 7. So, that reminds me of the Seinfeld episode where George Costanza decided he was going to name his child 7. But, George. All right, so this is a mature tRNA. tRNAs are not initially, they're transcribed to look like this. Okay, they have been modified, and they are greatly modified. And if one little misstep happens along that modification process, you would die. And so it's amazing everything that has to happen in order for the tRNAs to be functionally relevant. What do tRNAs do? Like, what's the whole purpose of having a tRNA?
Go between nucleotides and proteins.
Nucleotides and proteins.
Amino acids.
Amino acids and proteins.
No, nucleotides and amino acids.
Nucleotides and amino acids.
That's what I'm sticking with.
What?
Oh.
Okay, yeah, so this is sort of like the, I don't know, stenographer of the of the cellular world. This is the one that it's going to read the codon, and it's going to know, being anthropomorphic here, it's going to be able to equate what that codon is to what amino acid. So it's the go-between, the liaison, between the original transcript and the ribosome. Okay. And I want you to know the parts to this. So this end, this one right here, that helix, that P, it's called the acceptor stem. It's also called the aminoacyl stem. Because it's, here, let me just type out aminoacyl that way. Oops, let me do this color. That way you can see what I'm saying. Aminoacyl stem or helix. And that's because, remember, YL from organic chemistry just means it's in a functional group that's attached, and that's where the amino acid gets attached. It gets attached right here at the end of the CCA, and it has to be CCA. If it's any other nucleotide there, it's not going to work because, I mean, it won't. The enzymes don't recognize it, and so we'll get to that in just a moment. But it's also called the acceptor stem because it accepts the amino acids. Okay, so this one right here is called the D loop, and of course this would be the D stem or the D helix. And the reason being is the loop that's on the end of it always has to have one of its nucleotides, and it's the same nucleotide every time, that is modified. It's a dihydrouracil. Okay, and so that's why this is the D, as in day storms. Yeah. This is the anticodon loop and the anticodon stem that's attached, or the anticodon helix. And the letters, by the way, that are in there are those that are universally conserved. So if you notice, there's like 2 G's in that one. There's a U that's always by the wobble bases. By the actual anticodon. Okay. Now, the next one is this little nubby-looking thing. That's called the variable loop because it varies. Some of the tRNAs, it's just this little bulge. I want to say it's one of the phenylalanine ones. It's huge. It sticks way out. It protrudes way out. And so, that's why it varies. It's the variable loop. And then the last one is called T-Cy-C. And sometimes you just call it the T-stem and T-loop, but it stands for T-Cy-C. And thank you. The Cy, remember that was that pseudouracil, the pseudouridine. It's a specialty, and every tRNA has to be modified there, otherwise sound effects optional. Okay.
Size a special G.
Cy is a special uracil. Mm-hmm. Okay. And so I do want you to know that. And so this down here is going to be the anticodon. And whenever you look at it in 3 dimensions, it's not perfectly round like that. It really does go at an angle. So the one that's the highest one up that's your wobble base. Because remember, that's the one that allows for the degeneracy of the genetic code. Whereas the way it's written here, for those 3, it looks like they're all equally spaced, but they really aren't in 3 dimensions. And I do want you to be able to know the different parts of the tRNA. So this is now color-coded. Okay, so you can see, and I always have problems telling the colors apart, but in 3 dimensions, so this was the clover leaf, but then in 3 dimensions, the acceptor stem and the T-C stem look like they form one really long helix. See right through here? And it's because of the way that it has bent. And so they're not fully, they're like where my 2 thumbs are would be, they're not joined there, but it looks like 3 dimensions make the top of the 7 or the gamma or whatever, the bottom of the L if you turn it upside down. And likewise, the D loop and the anticodon, the D stem and the anticodon stem, they also form the other stick of the L. And then the variable loop would be sticking out one way or the other, just depending upon whatever the tRNA is.
So when you said the higher nucleotide, can you point that out on the...
So on here, see this one right here, this one's the bottom, and that one's the top, for lack of a better word. So when the tRNA comes and binds to the codon, the anticodon 2 of the nucleotides form really good bonds with it. But that third one, the one I say is the higher one of the 3, it doesn't form as good a hydrogen bonding. And so that's why it can be more flexible in whatever it is. It's some of the amino acids have more than one genetic codon. Okay, so that right there is the wobble base. The wobble base. And if you notice this is the 3' end, because this is a mature tRNA, so the 5' end has already been cut off, and this is the 3' end, and the amino acid is really tiny, but it gets stuck right there on the end. So compared to the tRNA, it's just this little tiny thing. And so there are various videos, and I can't remember if I've included a link to the one or not, that I try to show you I'm just amazed about how it all happens, and it comes together, and it has to happen so fast, and have high degree of fidelity, or otherwise, bad things would happen. This is the tRNA maturation pathway. It's the last thing I want to get through today. So in us, in mammals and those that have introns, this would be the primary transcript. It has a 5' leader and a 3' trailer. Okay? And many times it's got an intron there in the middle. So RNase P, ribonuclease P, this is the enzyme that I worked on. It has this one job, and that's to cut off that 5' end. And it's the very first step in the tRNA maturation pathway, and it cuts a specific bond, and it will always result in a 5' phosphate. If it doesn't, if it cleaves on the other side of the phosphorus atom of the backbone, you would die. You have to have a 5' phosphate. They don't really, whenever I was working on it, they don't know for sure why, but you have to, or otherwise it's gone. So that's why we were trying to develop antibiotics to combat the bacterial version of this, and you could potentially have a viable antibiotic where it would leave us alone, but it would kill off the bacteria because the bacteria couldn't make proteins ultimately. Then there are actually multiple endo and exonucleases that can cleave off the 3' trailer. And if that CCA was not in the original transcript, which many times it's not, there's a specific enzyme called, the full name is 3' tRNA nucleotidyl transferase. And all it does is it knows it's got to stick a CCA there on the end, or otherwise we'd be dead. And so now I think in bacteria, many times the CCA, I wouldn't say bacteria, many times that CCA is already encoded in the gene. But it's another checkpoint, because a lot of these are checkpoints to help regulate protein synthesis, because if we don't have enough tRNAs around, we won't be making the proteins. And so it's just another way that we have to keep everything in check. And then finally for us, the intron, the intervening sequence, this is a fancy word for intron, gets spliced out, and then we have our mature tRNA that can be amino acylated and go on to help out with the whole process of translation. Okay, so at this point,